Planet Earth has more than 1 trillion species, yet most of science has been focused on only a handful of so-called model organisms, like mice and flies. And within these organisms, scientists tend to focus on the parts that look most like humans. Take actin, for example. Actin is a part of the skeleton of the cell, and it works together with a bunch of other proteins to control cell shape and movement. And actin has not changed much throughout evolution, meaning that many different living things have actin proteins that look just like humans. For decades, scientists have been studying human-like actin, gaining more and more confidence about how it works inside of cells. But recently, Dr. Prachee Avasthi and her team made an insightful discovery that questions whether we really know actin as well as we think we do. Prachee's lab was studying a tiny green plant called Chlamydomonas, which swims around in water and soil. When scientists had removed the human-like actin from Chlamydomonas cells, they swam a little slower, but they were mostly fine, leading researchers to believe that actin simply was not important inside these tiny plant cells. But instead of focusing on what was similar about actin in humans and Chlamydomonas, Prachee's lab looked for things that were different, and they ended up characterizing a distinct form of actin that barely resembles the human one that everyone else had been studying. Surprisingly, the clammy-specific actin, despite looking totally different, can still manage to work together with the same protein regulators as the human one. Thus, when the human-like actin is removed, these cells are fine, but when both actins are removed, the cells can no longer function. Studying this alternate actin in Chlamydomonas has revealed that actin does a lot more than we thought, not only in plants but in humans and other species as well. At Arcadia, researchers are expanding the types of organisms we study and focusing on what's different about them to unlock biology's secrets about what we think we know and what we haven't even imagined yet.